on genome sequencing of thousands of patients with rare blood disorders.
Well, thanks very much. It's a pleasure to be here. Thanks to the organizers. I'm afraid I have a complete absence of graphics in my slides, so I'll rely on your imaginations. So it may surprise you, but actually about 3 million people around the world have a rare bleeding or platelet disorder. That's quite a few people. Now, you've just heard about hemophilia. That's one of the most well-known examples. But there are actually dozens of different disorders that involve abnormal bleeding. And there are many, many yet to be discovered. And you may wonder, well, is this only relevant for patients with these rare disorders, each one being individually rare but collectively affecting many people? And the answer is not necessarily, because platelets are actually involved in some way or another in a huge proportion of deaths in Western society. So if you look at heart attack and stroke, platelets are involved over there. So the more we can learn about how platelets function and the role in rare diseases of platelets, then perhaps we can learn something about these more common conditions. So the NIHR Bioresource for Rare Diseases in the UK, with many collaborators around the world, have engaged in a pretty large genome sequencing study. And the process is basically to collect samples and enroll patients from around the world with particularly rare disorders. One of them is the bleeding and platelet disorders subgroup, but there are other patients with immune deficiencies, pulmonary arterial hypertension, etc., etc. And the reason for putting together all of these different patients is that we can get uniform genome sequencing. So there's no batch effects. We can really get very nice data and at a very good price as well. So once we've collected the samples and we've done the genome sequencing, we also need to record all the detailed clinical features of these patients. So, symptoms, laboratory test results, whether there's a family history, because we're looking at heritable disorders. And so collecting the data and then recording it systematically in a database is one of the big challenges as well. And we've worked on particular types of coding systems to record these symptoms in a very systematic way. And the analysis, which is the bit that I'm involved in, involves looking at whether there are commonalities, so clinical commonalities between little subgroups of patients who also have commonalities in terms of their genetics. And when we do that, then hopefully we can discover novel causes of disease and potentially further down the line look at some treatments, of course. So far, this is an ongoing project, but we have already published a few papers on some new findings. So we've discovered a new genetic link between very large platelets, which are too few in number, and can result in some bleeding as well. And it goes hand in hand with deafness. So there is one gene which is quite well known, which is called MYH9, and that is the classic large platelet, few in number, with hearing loss, also kidney problems and so on. So this is really the second main gene in which you've got hearing loss and macrothrombocytopenia, which is this platelet disorder, going hand in hand in these families. There are currently 3 families which we know of, and the mechanism has been worked out to some detail. Another example that I can give you is a genetic change in a very well-known cancer gene called SRC. So SRC is actually the first ever discovered oncogene. You can find publications dating back to 1911. The first examples involved an avian flu virus, which contained this gene and then induced sarcoma in chickens. So it really dates more than 100 years. But until now, no germline, so no heritable variants had been found in this gene. And what we found was a gain of function variant in this gene, so it becomes hyperactive. And if you look at the symptoms in this family, what you have is very fragile bones with, for example, teeth fractures. You've got fibrosis of the bone marrow, low platelet counts, and some bleeding as well. A third example involves the gene called TRPM7. And this is a new genetic link between, again, large platelets and also heart rhythm problems. So, and in this particular case, because the mechanism has been worked out to some detail, it appears as if magnesium supplementation could potentially be some kind of treatment looking into the future. But importantly, apart from just finding novel causes of disease, we've also been able to extend the repertoire of variants in already existing genes that were already known about. But in some cases, there were only maybe 1 or 2 reports in the literature, and we've been able to expand that in some cases by 2 or 3-fold. So, for example, the actinin-1 gene, which causes macrothrombocytopenia again, so large platelets, few in number, CYCS, cytochrome C, that's another example, and many, many other genes. So this is also a sort of second benefit of this study. So just to conclude, I think that bringing together the genetic and the clinical data from hundreds of patients worldwide, and I stress worldwide, you need worldwide collaboration because rare disease patients are, of course, rare, and therefore you need to collaborate across the world to get the sufficient numbers to discover the cause of disease. So that's essential to discover new variants. It is important also to improve our basic understanding of platelet biology and perhaps bring benefits to the millions of patients with heart attacks and stroke. And another important point is that within months of publication, we can take these genes and incorporate them into a diagnostic platform. So this in the past would have taken years, but some of the genes that we've already published are already in our diagnostic platform. This is called the thrombogenomics platform. It covers all of the genes which are currently known to be involved in bleeding and platelet disorders. It's available to anyone in the UK, but actually also worldwide this year. We would really welcome anybody who has got a sample from a patient with a bleeding or a platelet disorder to send it across to us, and we'll be happy to do the targeted sequencing in the known genes. Thanks very much.
Thank you. Thank you very much. Any questions from the audience, please? I guess you had no time to go into detail, but how did you really prove that a certain genetic aberration relates to a physical disability? What sort of statistics?
Yeah, so we've had to use numerous different approaches, and it's not only based on the statistics, so lots of follow-up work has been required. So one essential thing to do is, of course, co-segregation studies. So you look at the family structure, who's affected, who's not affected. You have to make sure that only those who are affected carry the variant, and those who are not do not carry it in the case of dominant disease. But in all of these cases, there have been extensive follow-up experiments in the lab to make sure that that was exactly the mechanism. So there's been transfection into cells, and to look at the cellular phenotype and whether it can be rescued in various ways. So because the numbers are often low, it is very rare that you can just make a statistical argument and then get away with it and just publish it, right? So you get a candidate gene, if you like, or a candidate variant out of your statistical analysis, and then subsequently a lot of work needs to be done to make sure that you haven't got a false result. We've also developed statistical methods. So I've worked on a method which we call phenotype similarity regression, and the idea there is to look for similarities at the phenotype level between patients and to see whether those similarities can predict your genotype. So we've had to use a number of different approaches.
Right. Dr. McIntyre.
Could you just tell us what your thrombogenomics service is? Do you buy the diagnostic panel, the result, or is it a scientific collaboration between the submitting center, and can a patient submit his own DNA independently of a hospital accompanied request?
Okay, so the platform is a targeted, it's microarray based. So we have probes on a microarray. These probes target all of it's about 80 different genes. We cover all of the exons, many of the introns, many of the promoters. In the case of factor 8, which I believe you often have inversions, so in some cases we actually tiled the entire gene, so we can try to look for those. And the actual processing is done in Cambridge. The sequencing is also done in Cambridge. And what happens then is that the variants which have been called are uploaded onto a web application along with the phenotypes. And then the referring clinician, along with geneticists, bioinformaticians, can work together during what's called a multidisciplinary team meeting, analyze results together, and then generate a report. That report can then be sent back to the clinician, and then it's up to them what happens. So to answer the second question about whether a patient can submit his or her own sample directly, I believe that's not the case currently, but we're extremely interested in being able to reach more people by allowing a sort of more direct link with the patient. So it's something that we're actively exploring. The publication that relates to thrombogenomics involves 300 patients. It was published yesterday, I think, in Blood. And so far, I think we've reached about 600, and we're keen to reach many, many thousands, because the idea here is to be able to systematically collect the data so that we can get a very good idea of the spectrum of symptoms that you can get for each individual disorder. So there you go.
Well, my final question would be that relates to your second concluding remarks. Maybe you can be a bit more extensive on that, how these basic biology findings might benefit the patients. Can you give an example?
So, in the case of the rare disease patients, so let's take the example of SRC. Okay, so SRC is a cancer gene. There are currently numerous SRC kinase inhibitors which are being trialed, and therefore, if you have a patient with an overactive SRC protein, it makes sense to trial an inhibitor to see if that can help. Now, of course, actually, it turns out that a side effect of some of these inhibitors is bleeding, which is one of the problems that the patients in this case have. So one has to be very, very careful. But that's one example of how that could benefit the rare disease patients. In the case of TRPM7, it has to do with a magnesium ion transporter. So one hypothesis is that magnesium supplementation could help there as well. So when the mechanism is worked out, then of course that kind of benefit could occur. But there are other benefits which are to do with being able to tell whether there is some predisposition to some other condition. So there are many bleeding and platelet disorder related genes where you have also predisposition to leukemia. So if you know that your variant that causes your thrombocytopenia is in one of those genes and not in one of the other genes that don't predispose to leukemia, then you might say, well, in your particular case, perhaps we ought to go for some regular screening or something of that nature. So I think the knowledge on its own can benefit patients, even if there's no direct treatment.
Dr. Hayu, have a final question?
Do I?
Yes, well, if you have a question, please go ahead. You're an expert in the field.
Okay, well, I was actually curious if you could tell us something about what do you see as the unifying defect at the cellular level between large platelets and congenital deafness? Do you have any idea? The DIAPH mutation?
Yeah, so why the mechanism underlying the two? Well, it's all to do with the cytoskeleton in the case of the platelets. I think that connection with the hearing loss hasn't been fully worked out, but it's already a second example where those two conditions go together. So I hope that can be worked out eventually.
Well, thank